DNA replication. The process by which DNA duplicates itself is called replication. Through replication, it forms two copies that are identical to it. In eukaryotic organisms, replication of DNA takes place only once in the cell cycle. It occurs in the S phase of interphase in the cell cycle. DNA replicates through semi-conservative mode of replication. Means, in both the daughter DNA molecules, one strand is from parent DNA and another strand is newly synthesized. Activation of nucleotides. 4 types of nucleotides of DNA, that is deoxyribose AMP, deoxyribose GMP, deoxyribose CMP and deoxyribose TMP are present in the nucleoplasm. They are activated by ATP in presence of an enzyme phosphorylase. This results in the formation of deoxyribonucleotide triphosphate, that is DATP, DGTP, DCTP and DTTP. This process is known as phosphorylation. Point of origin or initiation point. It begins at Pacific Point O. It's a point where DNA replication takes place. It is also called as replicon. In eukaryotes, there are several replicons, several points of origin. In prokaryotes, there is only one replicon. At the point of origin, enzyme endonuclease nicks cut one of the strand of DNA temporarily. The nick occurs in the sugar phosphate backbone or the phosphodiester bond. Unwinding of DNA molecule. Enzyme DNA helicase operates by breaking weak hydrogen bond in the vicinity of origin. The strand of DNA separate and unwind. This unwinding is bidirectional and continues as Y-shaped replication fork. Each separated strand acts as template. The two separated strands are prevented from recoiling, rejoining by single strand binding protein. SSB protein remains attached to both the separated strand so as to facilitate synthesis of new polynucleotide strand. Replicating fork. The point formed due to unwinding and separation of two strands appears like a Y-shaped fork called replication fork. Synthesis of new strand. Each separated strand acts as a mold or template for the synthesis of new complementary strand. It begins with the help of small RNA molecule called RNA primer. RNA primer gets associated with the 3' end of template strand and attracts complementary nucleotide from surrounding nucleoplasm. This nucleotides molecule bind to the complementary nucleotides on the template strand by forming hydrogen bond, that is adenine double hydrogen bonded with thymine and vice versa, guanine triple hydrogen bonded with cytosine and vice versa. The newly formed nucleotide get interconnected by phosphodiester bond forming a polynucleotide strand. The synthesis of new complementary strand is catalyzed by enzyme DNA polymerase. The new complementary strand is always formed in 5'3' direction. Leading and lagging strand. The template strand with free 3' end is called leading template and with free 5' end is called lagging template. The process of replication always starts at 3 end of template strand and proceed towards 5 end, as both the strands of the parental DNA are anti-parallel. New strands are always formed in 5-3- direction. One of the newly synthesized strand develops continuously towards replicating fork is called leading strand. Another new strand develops discontinuously away from the replicating fork is called lagging strand. Maturation of Okazaki's fragment. DNA synthesis on lagging template takes place in the form of small fragments called Okazaki's fragment, named after scientist Okazaki. Okazaki fragments are joined by enzyme DNA ligase. RNA primers are removed by DNA polymerase and replaced by DNA sequence with the help of DNA polymerase 1 in prokaryotes and DNA polymerase alpha in eukaryotes. Finally, DNA gyrase topoisomerase enzyme forms double helix to form daughter DNA molecules. Formation of daughter DNA molecules. At the end of replication, 2 daughter DNA molecules are formed. In each daughter DNA, one strand is parental and the other one is totally newly synthesized. Thus, 50% is contributed by mother DNA, hence it is described as semi-conservative replication.